station. It's an
Yes,
open
or
discussion.
you can put it on the chat.
Yes, maybe, but in general, when I present, I don't see the chat or.
Yeah. No, but if needed, I can read the question to you.
Okay, perfect. Welcome to everybody. My name is Frédéric de Haran. I work at the CRW in Belgium. CRW, it's the Walloon Agricultural Research Center. It's based in Jean-Blou. And my expertise is to work with mid-infrared spectroscopy to develop new models to help the farmers and the milk recording organization or other people to help the farmer to well manage their farm. And the purpose of my presentation is to explain why mid-infrared spectroscopy could help you to improve or better understand the resilience of the dairy sector. Just a moment. Okay. When we speak about resilience, and mainly in the research for dairy project, we focus on 3 knowledge areas in order to enhance dairy farmers' sustainability. And these 3 main areas, it's technical efficiency, economic social resilience, and environmental animal welfare and society-friendly production system. But to well understand these 3 main focus, if we want to understand and effectively manage resilience of the systems, in general, it requires having a large amount of data or measurement that accurately represent their complexity, diversity, and evolution. The problem is always the same. If we want to have a lot of data, if possible, it will. We want that this data acquisition are not too expensive. Okay, let's focus more in detail on this phrase. We need a large number of data measurement and not too expensive. That's why we were interested by this approach of mid-infrared, because during the milking, we have different steps where we collect samples. The first ones during the milking at the milking barns, for example, the milk recording acquisition samples, or during for the milk payment during the collection of the milk at the farm for the milk payment. And also we have also samples collected during the process of the transformation at the industry levels. And all this these samples are in general analyzed in the milking labs by techniques called mid-infrared spectroscopy. And I will explain a little bit more what is mid-infrared spectroscopy. Mid-infrared spectroscopy, it's a technique. We will use the infrared region. It's a region between the region of light and the microwaves region. And this region, it's in 3 parts. The first part is the near infrared region, the mid-infrared region, and the far infrared region. And in my presentation, I will focus more on this mid-infrared spectroscopy part. because it's important in milk analysis. In this presentation, I present here what we call a spectra. A spectra, it's the combination of the different absorbance in the region of the mid-infrared, covering the whole part of the mid-infrared region. And this spectra is very important in mid-infrared analysis because we could use some parts of the spectra to create models to predict qualitative and quantitative models. When we say qualitative, it's because some part of this spectra give a very good, very precise answer to the detection of different bounds. In this example, the CO bounds in the milk, or the CNNH bounds, or here the CO and OH bounds. And these bounds are directly related with the composition of the milk. For example, here the CO double bonds are directly related with the fat because the CO is only present with the carboxyl part of the fatty acid. It means that with one spectra we are able to give to say that we have presence or not of fat, just due to the fact that we have presence of the CO bounds. But also it's the techniques to give to us the possibility to have a quantitative approach analysis, because the absorbance, the level of the absorbance of this peak, it's directly related with the concentration of these bonds in the milk. That's why this methodology is used in general for the payment, because with one model for the prediction of the fat, for example, with one model, with one mathematical model, we are able to predict the fat, the protein, and so on. The aim of the project, of the different projects in the CRW, is to create new models, not only fat models or protein models, but new models to help us to predict what we call direct models, indirect models, pseudo-indirect models, or other models. And we will go through these different models just to show, to illustrate how we can use these techniques to help you to well manage the resilience of your farms. But all these models that we have created in the past have their own quality. Not all the models are very good, but some are poor or are relatively good. And to estimate the model quality, we realize what we call clusters. It's based on the clustering of different statistical statistics of the models. It's the ratio performance of deviation, the root mean square error of the models, and the R square of the models. And here it's represented the different quality of the models. In green, it's a very good models, and in red, the poor models. But just have in mind that the color will define the quality of the models, And sometimes, even if we have not a green models, it could be very interesting to use it because we will have models who are able to differentiate it to differentiate the high or the low levels, and it could be interesting. But we will go with some example to illustrate what I want to say. The direct models, in general, are models based directly on the composition of the milk. As explained before, for example, fat, protein, lactose are components in the milk with concentration higher than 100 ppm. And in general, these models are very good. Its model quality are very good. It's more precise. In general, we say that the mid-infrared spectroscopy is more precise than the reference analysis because the precision of this method is very, very, very good. That's why it's used in general in payment. But after that, we have developed it in beginning 2006. We have developed first models to predict what we call a fatty acid component. And it was based on the first publication here. We had first publication here is the second publication that we made in 2011. But this model is very important because in general, when we use major fatty acid components, you are able to give more details in the composition of the milk. It's interesting to give information for nutritional quality of the milk, but also this fatty acid composition have a big influence on physical properties of the milk. For example, for the butter, the butter quality and so on depends of the composition of the fatty acid, but also and in this presentation I will give some example, but the fatty acid composition also give co-status. It's a good co-status biomarkers, for example, for energy balance or rumination. Here is the representation of the different models created for the fatty acid. As you can see, we have a large number of models created with relatively good quality. We have a lot of green models and some yellow, not a lot, and we have no red quality models. And in general, the quality of the model depends of the concentration of the fatty acid. If we have a low concentration in terms of fatty acid, in general, we have a poorest model quality. Higher is the concentration, higher is the model quality. And as I said, fatty acid, it's interesting. And just to illustrate my purpose, for rumination, for example, it's interesting to use the fatty acid because the fatty acid have two origins in the milk. As you know, as you maybe know or not, I don't know. The first, we have a first big part of fatty acid, what we call the de novo fatty acid. It's the fatty acid that comes directly from the feed, and during the transformation of the feed in the rumen fermentation, we have just a minute. We have formation of acetate and butyrate. It's a volatile fatty acid in the rumen, and this volatile fatty acid give directly the de novo synthetic, the de novo fatty acid. In general, the de novo fatty acid have a fatty acid fatty acid distance less than 14 carbon. And the second family it's the mixed and performed fatty acid synthesis. This fatty acid come directly from the feed, the fatty acid present in the feed. And it's in general we accept that this fatty acid family is fatty acid with fatty acids the longer the carbon chain have fatty acid larger than higher than 18 carbon. And if we have more, what I want to say, if we have more de novo fatty acid in the milk fat, it means that the rumination is better. If we have less de novo and more mixed performed fatty acid, it means that the rumination is not very well. That's a good indicator of the rumination. I'm not sure that I was. If you have questions, please do not hesitate. Okay. Just a minute, because I want to close my laser pointer, but I have problem to. Oh, I continue. Second use of the fatty acid composition could be, for example, here it's a new paper that we have published last year. It's the ability to use the fatty acid to discriminate the presence or not of grass in the ration. It's a good aspect. With the fatty acid, we are able to say if a cow have a large quantities or a low quantity of grass in this ration. Let's go to continue with other models. The second family is the indirect models. And just to illustrate what I want to say, for example, we are able to create models to predict the methane emission. We have made a big project called MetaMilk. And in this project, we have built models to predict the ejected methane. To do that, we have measured animals with the methane emission. And we have taken samples, milk samples, and we have created equation to predict the methane. And as you can see here, the model quality it's not very good, but it's usable to detect animals with low or high potential to emit ejected methane. And it's interesting, for example, if we want to detect animals, a big emitter or low emitter. And for genetics purpose, it's very interesting because in general, research, we have a lot of data to detect these animals, and it's a good way to have it's a good way to have genetic tools to detect these animals. Unfortunately, I have not a lot of time to explain well in details, but if you have questions, please do not hesitate. Another aspect, an indirect models, it's the health status models that we have created, and I will give you some models that we have created. First example is the cow status and the presence or not of mastitis. As you know, in general, we are able to measure the presence or not of mastitis by the way of the cells count, but we search to detect some components that can have a more rapid answer to the presence of mastitis. That's why we focus on the, for example, the lactoferrin. The lactoferrin, it's a glycoprotein, very important because in general, the lactoferrin, if the lactoferrin is high, it means that something are not working well in the cow, and it's a good indicator of maybe a potential mastitis who will be developed in the cow. That's why we had focused on the lactoferrin concentration. As you can see here, also we have a quality models not very good, but with this type of models we are able to detect a potential cow with problem. Another parameters, it's the NAGAS parameters. Here also we have a very, in this example, we have a very bad quality models, but we have developed not because here in this first example, it's the concentration. It's a model based on the concentration. It's not good. But if we create another model to say, and in this model we try to separate high and low, and if we try to predict high and low to discriminate the high and low, with these models, we are able to have a good results. We have better results with these discriminant models, and we are able to discriminate high and low NAGAS concentration of the cow. Another example of cow status, of health status, is the energy balance cow status, and we have two types of models. The first one is the models who are able to predict milk biomarkers in relation with energy balance. And in the first table here, we can see that we have different biomarkers in the milk, and more and these models are with a low or high quality, and especially we have milk citrates, which could be interesting biomarkers to detect the energy balance. The other one not works, unfortunately. But also we try to measure blood biomarkers through the milk spectra, and it works for the blood IGF-1. As you can see, we are able to discriminate high and low cows with these blood biomarkers with IGF-1. That could be interesting. And the ketosis, it's another health status example that I want to present. The ketosis, we have also different models here. And as you can see, with the milk BHB, in concentration could be an interesting models. But if you use these models in discrimination, the qualitative models, we have better results, and we are able to discriminate high and low cows. Just to illustrate, I also put some examples with the blood biomarkers and the presence of blood BHB. And as you can see, we have similar results that the BHB directly in concentration in the milk. Another example is the nitrogen efficiency. We are able to predict the nitrogen use efficiency by the use of the spectra. It could be interesting because the nitrogen use efficiency is more and more important, and to detect animals with low or high nitrogen use efficiency. And the spectra is very interesting, but this is models who are continue. We continue the works because we have a low number of data in this aspect, but we continue the works because it's more and more interesting. Now, another aspect is the detection of animal stress, and more. I want to present more an example regarding the chronic stress detection. Alors, concerning the stress, in general, you have acute stress or chronic stress. And the acute stress, it's in this graph, it's here. We just have a stress during a short time period. And in general, the animal have a good resistance to this stress. The problem with the stress, it's when we have a continuous stress. In general, the animal have a good resistance in the beginning of the continuous stress, but after a period, we have an exhaustion of the animal, and if we have this exhaustion, the problem, the resistance on the stress, it's lower because the animal is exhausted to the stress, and we have some problem who will appear. For example, the susceptibility to metabolic, inflammatory, and infectious disease, or maybe we can have fertility troubles or growth disturbance. We have a loss of weight, or the milk production could decrease with this stress. That's why it's important to have to detect this stress, because the production and the economics of the farm will decrease, the welfare of the animal will decrease, and the societal perception of the dairy production also will decrease. If we have a lot of animal stress in the herd, it could be a problem. The problem, we have no consensus in the literature regarding the chronic stress biomarkers. That's the problem. That's why we have organized in the past an experiment. And in this experiment, we have detected two potential biomarkers. The first one is the air cortisol concentration. The air cortisol, the cortisol in general, it's a good standard chronic stress biomarker. And to do that, we collect in the tail of the cow, and here in green, in the lower part of the tail of the air of the tail, in this part, in the 1.5 centimeters, it's because the tail of the hair of the tail increase all the time, growth on all the time, and the concentration of the cortisol in this part, it's directly related with the cortisol in the blood during the last part of the life of the cow. That's why we are able, by measuring the concentration of the cortisol in this part, the last part of the growth of the hair, we are able to detect if a cow is in stress or not. Another potential parameters to detect the stress or not, it's the blood fructosamine. It's relatively easy to collect. It's just the collection of blood samples, and we measure the blood fructosamine. Okay, now as we have detected these two potential parameters of stress, of chronic stress biomarkers, now we will see if we are able, by using the milk samples, to create models to measure the presence of air cortisol or the blood fructosamine. To do that, we have organized a large-scale samples protocol. We have collected a large number of samples in different countries, in different Earths, in covering all the parities, all the lactation stages, covering all the seasons, all the areas, and so on, and also all the breeds. And we give to the all farms in these experiments, we give some recommendation to collect these samples. We ask to them to collect, to try to collect cows with good welfare, without stress potential, and also to try to collect cows with potentially suffering with stress, a chronic stress. What is a chronic stress? It could be overstocking, chronic disease, permanent source of fear or pain, dependent of some aspects, environmental discomforts, limited access to food or water, or inadequate barn design or location. It's always difficult to find or to be sure that an animal is stressed or not. But as we have collected the milk, but also the air of the tails and the blood, and by measuring the cortisol and the fructosamine, we will have the answer if an animal is potentially stressed or not. Okay, now we will focus on the models created for the cortisol discriminant models. Here it's the representation of the distribution of our data, and the first part was to find the threshold, because we need to find if an animal is stressed or not, and we need to fix a threshold. It was the first part. We have fixed the threshold to 22 picogram per milligram, and we say if we have higher level than this threshold, we have potential chronic stress. That's the way we proceed. And here it's the discriminant models statistics. And as you can see, we have made a cross-validation and also an external Earth validation. And we will focus only on the second part, because it's the more important. The external validation gives this information. It seems that we are able to discriminate high level of cortisol, and by this way, chronic stress, because we obtain relatively good results. We need to have more data to be sure that it works, but these first results are very interesting. Now, concerning the second chronic stress biomarkers, is the blood fructosamine. In this case, the blood fructosamine gives two answers, not only one, not only with the high concentration the chronic stress presents, but also with low concentration. We know that with low concentration, It means that with low concentration of blood fructosamine, it means that we have maybe an undernutrition or an energy deficit. It's more complicated to manage, but we try to do that, and we have created first to create a model to detect the undernutrition energy deficit, and we have fixed a threshold to 200 micromole per liter. And the first model that we created give these results. It seems that we are able, by using these models, our models based on the milk spectra of the milk, we are able to detect animal with undernutrition energy deficit. It's very interesting. And the second part, with higher threshold, with higher concentration, with a threshold of 230 micromole per liter, maybe it gives answer to detect animals with chronic stress. And the models here, it's give also interesting results, because we have relatively good sensibility, specificity, and accuracy, and it seems that we have also models able to discriminate this presence or not of fructosamine and potentially stress animal. And now, if we combine the two models, the high and the low models, with the combination, we are able to detect if animal is with under and high stress potential. Okay, I go rapidly on this other families of models. It's the pseudo-direct models. It means that we try to predict parameters with lower concentration in the milk. It means with concentration less than 100 ppm. It means that it's not a direct models, because we are not able with the mid-infrared spectroscopy to detect this very low concentration, but with maybe relationship in the milk. And for example, just to illustrate what I want to say, for example, minerals, it's not an organic bond, have no organic bond in reality, because in the beginning, I say that mid-infrared spectroscopy is very interesting because we have interaction with the organic bonds such as CO, CH, and so on. But with minerals, we have not this kind of organic bond. It's minerals. But the calcium, for example, will have interaction with the protein, for example, and with this interaction with the protein, they will change the structure, the structure of the protein, and we will see with this interaction. We will see this interaction because they change a little bit the structure of the protein. Not easy to explain in 2 minutes, but I'll try. Okay, and the minerals, it's very interesting to focus on, because it gives information on nutritional quality, but also on different technology properties. And for example, it's very interesting during the cheese process. The minerals also give information on the co-status biomarkers, for example, the mastitis. We know that some minerals are good indicators of mastitis. And also, but especially in Holland, you know that eutrophication issue is very important, and the presence or not of phosphorus could be interesting to predict. And in the statistics of the different models that we have created, you can see that we have relatively good models, not for sodium, but for the other one, we have relatively good models with relatively interesting models. Okay. And it's just an example to illustrate what we call others models. Others models, it's to detect abnormal spectra. In general, we have models quality who try to detect abnormal spectra. It's not used in Europe in general, these types of models. It's more used in India or something like that. It's in this country where they try to put other things than milk in the to try to increase the fat or the protein. But it's just to illustrate that we are able with the spectra analysis to detect abnormal milk. And in this example, it's a paper that we have done during the big problem in China, where they put melamine in the milk to increase the protein level, and we had shown that we are able to detect melamine, because the reference analysis don't differentiate the nitrogen, but in the mid-infrared spectroscopy, we are able to differentiate this melamine. And melamine, it's not natural nitrogen. It's not a protein. That's why we are able to detect this. But it's just to illustrate. But also we are able to predict, for example, detection of other components who are not, such as chlorates in the milk and so on. Not antibiotics, unfortunately. We are not able, because we are not able to detect antibiotics with these techniques. But okay. Now, if we go back to our first phrase, in this phrase, there is an important word. It's standardized data measurement, because standardized measurement is very important, because we don't want to compare apples and pears. It's very important always when we do analysis, reference analysis or mid-infrared spectroscopy, we need to be sure that the concentration that we measure is everywhere the same. That's why some international organization exists at national or international level. In general, you need to calibrate your apparatus, for example, for fat, protein, and so on. At international or national level, we need to calibrate to be sure that we have all the same levels. But for some parameters, this solution of calibration doesn't exist. For example, fatty acid, we have no common way to proceed. That's why some projects start to think to this problem. The Extramir project, it's very new in ICAR and IDF. We try to find a way to be sure that the analysis of fatty acid are done everywhere at exactly the same levels. That's why we start with the fatty acid. But I had made the I tried to explain other models such as I don't know, methane, nitrogen use efficiency, and so on. In this case, it's not possible to realize calibration of our models, because when you want to calibrate an apparatus, a mid-infrared apparatus, in general, you measure samples with different level of concentration. And just for example, the fat, you receive different different samples with fat concentration increasing or decreasing. You measure these samples with the reference analysis, and you compare this. You compare what you obtain with the mid-infrared and with the reference analysis, and you correct your apparatus. That's in general how you proceed. But for the methane emission, for example, we are not able to do this because it's an indirect measurement, and you are not able to have a sample set of milk with different methane emission. That's why we need to find another way to proceed. And it's a big work that we have developed in the CRW. It's what we call the mid-infrared spectral standardization. In this project, we will standardize directly the spectra. I will explain a little bit more in detail. But first, why it's important? Because we need to harmonize the spectra across the different instruments, labs, countries, continents, because some labs use one brand models of apparatus, other brand models apparatus. And if we want to share models, we need to be sure that every apparatus gives exactly the same spectra. And also, it's important if we want to share data, and I will go back to this problem. But when we try to develop models, we need to share a lot of data, or reference data, and a lot of spectra. And we need to be sure that everybody works on the same levels. That's why it's important. Also, the instruments will deviate during the time. If we want to use a model during the time, we need to be sure that we have no deviation. We need to be sure that we have the stability of the prediction. And also, I spoke about the fact that we try to use the models predicted to use it with the genetic evaluation, for example, but we need to collect and to create a historical database, and to be sure that our historical database is always on the same level. We need to be sure that we have no fluctuation during the time, and that's why the standardization is important. And how we proceed, we send sample set of different milk in the different labs, and we use a technique that we call piecewise direct standardization, PDS standardization method. And with this PDS method, as you can see here, it's the representation of the PCA of the informative weight numbers of all samples that we send to it's 5 common samples analyzed on 45 instruments from the same brand. And then, as you can see, we have in blue, with no standardization, a large variability of this spectra. It means that it's not possible to use any models on this spectra. But after standardization in green, as you can see, we are able to decrease this variability due to the instruments or the instrument difference, and we are able to decrease this variability. It means that by using this method, we are able to use common models with the different instruments. We have made this project with the EMR, the European Milk Recording, and the CRW is in charge of the organization of this standardization. The EMR is an organization regrouping different milk recording organizations in Europe, and it's in blue. In general, it's in this part. We have now in routine 37 labs who are participating to this standardization networks. It means that we have 112 apparatus, but also in grape, some countries participate to this standardization of their instruments because we have some exchange depending of different research projects. We have some exchange to be sure that when we create a common models, we works with standardized spectra. That's why they sometimes they participate or not to the standardization of instruments. Because, and when we go back to this phrase, when we collect data, we need to be sure that in our models we cover the whole variability existing in the reality. We need to be sure that the data that we collect accurately represent their complexity, diversity, and evolution. Because in general, when we create a models, we need to be sure that we cover the whole variability, because extrapolation is not. We can never use extrapolation samples. In this example, for example, in the red, the variability is covered, but if we put the red here, we never can be sure that here the predicted value will be the good one or not. It's with mid-infrared, it's not. We couldn't make like this. It's not. It's forbidden. It's forbidden. That's the word that I want to search. That's why it's very important to organize large collection of data coming from different countries, covering different brands, region, and so on. And that's why we have a lot of different collaboration at national and international level to collect this data, this reference data. For example, the methane emission models, to collect data from experimental farm, it's very expensive, very, very expensive. That's why collaboration is very important to have. And now we are able, in national level, in Belgium and more specifically in Wallonia, we have created what we call the Futurospect database. It's a standardized database of spectra, collecting the old spectra coming from two ways. The first way is individual spectra coming from the milk recording organization, and now we have a database of 7 million spectra. And also we have created an historical database with bulk spectra coming from the payment. And by this approach, we are able to cover different year, different region, different Earth during the whole time. It's very interesting to have this database. And we start in 2023 with the Interact project called Olico. We are able now to have a database, a European database, regrouping more than 60 million individual spectra. And with database, and it was the last point of my presentation. With this type of database, we will be able to understand and effectively manage the resilience of our systems. And 3 current areas of research are done here in CRW through 3 different projects: the Wally Smart, Olico, and Decide project. The first one it's a project called Wally Smart. The goal of this project is to establish a network of gathering farm data from diverse stakeholders and collectively processing it to derive maximum actionable insight for decision making. We want to collect data from spectra part, from the routine lab, but also data coming from vet information, from genomic evaluation, and so on. We want to collect and to create a platform. Or also we have information coming from meteo information, and to detect if we are able to have stable models during the time and with different climate change and so on. It's the first project. And a second one, it's called Olico. And in Olico, as you have seen, we have a lot of different models, and the main problem with the different models, it's a lot of data, and very difficult to manage for a farmer or even for technicians. It's too much data. That's why we have this type of project. It's to have we search to have an holistic approach. An holistic, it's with a lot of different data to search, to have decision-making tools in 6 different areas. The first one, it's concerning the health or the welfare. My cow is in good condition or not, a potential health problem or not, and maybe which one, but we can go step by step. Also, we have a second area. It's concerning the transformation. With this milk, can I use this milk to make my cheese or to make butter on some and this kind of answer. We need to have an easy way to answer to the farmer. The third one is the fertility aspect. Also, we need to find parameters to have an holistic approach for the production aspect and the environment aspects, and especially for methane in our mind. And also the heat stress, it's very important because due to the climate change, we have more and more problem of potential heat stress, and we need to focus on this important aspect. And the last point is the DSEED project. The DSEED project, it's a Walloon region project who tried to make life cycle assessment to evaluate the farm carbon footprint and the ammonia emission and also the energy consumption. And the spectra approach is more and more important and will be an integer in this life cycle assessment. That's an objective of this project DSEED. This will finish my presentation. If you have questions, please do not hesitate to answer me. I know that it's a lot of, just a moment, it's a lot of information received, but we have time, I think, to discuss, and no problem for me to, no problem for me to exchange on that. And I see that Amélie van Liert is here. It's our specialist in methane emission. If you have more question on the methane, please do not hesitate to ask to Amélie. No question? It was so clear? I'm supposed not. Hello, Amélie.
Hello.
If you want, you can write the question on the chat too, if someone could speak on. So I don't think there is any question. So, but thank you, Frédéric, for this presentation. It was really interesting.
And please do not hesitate to come with your question, maybe after by send me an email, or if you want to have more details on one aspect or not, do not hesitate. And maybe something very important, we are always open to any collaboration, because as mentioned in my presentation, collaboration is very important. We need to have access to different reference analysis, and all the time it's not easy. If you organize, for example, if you organize an experiment, or if you have in your farm some specific animals with presenting maybe a stress, I don't know, why to not exchange data and to be sure that we cover your variability in your farm? We are open to this type of collaboration. As I mentioned, I show that potentially we have shown that these types of tools can work, but the robustness of these models, it means when I say robustness, it's the potential used in routine. We are not sure that it works, because we are never sure that we cover the whole variability, but we are sure that potentially we have something that can be used in routine. But to do that, we need to be sure that we have robust models, and to do that, we need to have data. And to do that, we need to have collaboration.
Okay, thank you. I will stop the registration of the webinar.
Okay.